So good morning, good afternoon, or good evening to anyone. And today we're going to have another edition of our innovation stories. And today we're lucky to have Dr. Christine Carson, who is a CSO of CYTOPHENIX. So thank you, Christine, for joining us today.
Pleasure to be here, Munya.
Great, and I know that's just quite late on your end of the world, so really appreciate you making the time to be with us. So we'll just jump straight in. Um, I think these stories we always like to start from the beginning. So I don't know if you want to briefly introduce yourself, tell us kind of where you're from, what you've been doing, kind of prior to CytoPhoenix, and then maybe give us an idea of the type of problem that CytoPhoenix is trying to solve.
Okay, you might have to remind me of some of the question subparts as we go through. So, Dr. Christine Carson, I'm from Perth in Western Australia, in Australia, and prior to CytoPhenix, I was a career research microbiologist. I started out by going to university, the University of Western Australia, and I did a science degree. I hung around and did an honours year, so it's a postgraduate year, and we get to do a research project in honours at UWA. And I fell in love with the research process and I never left. So the honours turned into a PhD, which turned into a job, which gave me a heap of experience in a broad range of microbiology fields. And that eventually led me to working with a group who had invented a rapid way to assess antimicrobial susceptibility, so how susceptible microorganisms are to a particular antimicrobial agent. And that has led me to CyTOPHENIX. So it's been quite a journey so far.
Great. No, thanks a lot. And I think you've kind of hinted a little bit of kind of what CytoPhoenix is trying to solve, and I think it's around detecting microbial resistance. But would you just maybe just tell us a little bit more kind of about that aspect
Yeah,
and how you're kind of
certainly.
going about doing this? Yeah.
And it's very good timing too, because this week, starting today and for a whole week, is World Antimicrobial Resistance Awareness Week, or WAR for short. And it's a week where we try and raise awareness about the fact that many microorganisms that could cause infections and were previously readily treatable with antibiotics or antifungal agents or other antimicrobial agents. They've developed resistance to those agents, and sometimes we are now left with infections that are either very difficult to treat or, in fact, untreatable. One of the diagnostic tests that gets done routinely in clinical laboratories is called an antimicrobial susceptibility test. And let's say you have an infection and the microorganism that's causing that infection is cultured in the laboratory. They will actually take a little sample of it, reculture it in the presence of a whole range of potential medicines, and they'll work out which medicine would be the best for treating your infection. That's called an antimicrobial susceptibility test, or AST for short. And those tests can take 2 to 4 days, depending on how many tests you need to run, and how complicated the answer is. Well, our group has invented a way to do that test in 4 hours. So instead of it taking days to be able to have the evidence to help get you on the right treatment, that evidence can be available in a matter of hours. And we are very keen to bring that test to market so that more people can benefit from that kind of speed.
Oh, that's really fantastic. And I think yeah, really potentially a game changer. So thanks for that overview. And I'm quite interested in something that you said kind of at the beginning, kind of talking about your journey, and how kind of maybe an academic went from, academic science and kind of working their way into a startup. So would you mind kind of maybe telling us, had you always wanted to start a company, or is this something that kind of came along as you kind of worked with these individuals who had discovered this interesting technology?
Yeah, I wouldn't say that I had always wanted to start a company, but I'm certainly loving it now. And yes, so I'm really enjoying what I'm doing now. I was fortunate in that for a long time, my research was co-funded by industry and government. And so there has always been an answering the questions that industry needs answered component to the research work that I do. So I've always done very practical research, and that's probably stood me in good stead and set me up really well to be in the position that I am now, where, we need to demonstrate that our test does a particular thing. We need to prove it unequivocally and assess its performance and prove to other people that its performance is genuinely as good as it is, and then be able to put that test in the market. So I've long had an understanding of the kinds of things that industry needs from research, and now I'm really putting that into even more practice with my own company. So it's good.
Great. No, that's what that's going on. I think that kind of leads into like, I think an interesting question that I had here about validation kind of these early prototypes. And I think you did mention this kind of close relationship already with industry. I kind of want to know, how did you kind of go about validating some of these technologies? And really you've touched a little bit about this, but maybe expand a little bit about the partnerships with industry and how that kind of have helped company develop these technologies.
Certainly. So it's one thing to be able to make academic observations in a, in an academic setting about how well you believe your test works. But it's another thing to understand whether the workflow for that test will be practical in a clinical laboratory. I look back on some of our earliest workflows when we were really just discovering the test. And they're a long way back, but there's no way that those workflows would work in a clinical laboratory. A clinical laboratory, it's fast paced. There's many specimens coming through the door every day. There's an urgency about getting the results out. People are usually short-handed. The more you can automate, the better. So, we've worked really hard to make sure that our test will be practical in a clinical laboratory. And we've done that partly by having the right team. So some of our team members are experienced in working in clinical laboratories, and they've been able to advise us about, what will play in a clinical microbiology laboratory and what won't. We've also worked closely with clinical laboratories so that they can give us their unbiased feedback about whether or not that is the type of test that they would adopt. Would they consider using a test like ours? Does that workflow fit with what they do? So you really do need to work quite hard at building partnerships with all of the stakeholders that you are hoping to serve with your final product. Companies that try and develop particularly lab tests without consulting the end users in great detail and frequently do so at their own peril. Like, it's really important to talk to those end users, to get them involved, to have them road testing your test along the way.
Great. No, that sounds fantastic. And I think kind of still kind of in that area, during your development, can you think of a time when you realized that this particular idea that you had resonated with kind of the end users? I think you kind of talked about this back and forth with end users, but were they certain either milestones or indications that actually, the thing that you're working on in the lab seemed to actually provide value to the end users?
Yeah, look, it helped that one of our team is an infectious disease doctor in a clinical laboratory, and he was part of the development of the product. We also had recent medical graduates who were interested in microbiology and were working in microbiology laboratories. They would come through our academic lab doing particular projects, and we would get their input and feedback as well. And lots of them had rather impolite things to say about some of the incumbent systems. So, the incumbents they are the workhorses of antimicrobial susceptibility testing in the labs, but they use old technology and they're slow. And there are some combinations of antibiotics and bacteria that they don't do very well. And so you kind of end up with this patchwork quilt with a few holes in it of what the current incumbent technology can manage in the laboratory. And then people in the lab have to fill those gaps. Whereas if you have a test like ours, we haven't met a drug bug combination yet that we can't do. So, it's truly a universal test. The principles that apply in the performance of our test apply universally across all drug bug combinations. If you can grow it, we can test it. So it was important to have that feedback along the way. What features of our test interested medical and laboratory personnel who were actually working in clinical labs.
Mm. Yeah, thanks. And I think that leads me to another question. And, with these partnerships, they potentially are maybe areas where there might be friction as people kind of may have different expectations of particular partnerships. And I want to maybe probe a little bit, how did you manage those particular expectations? On one hand, you're kind of coming from a little bit of an academic background. There's also industry players, and then now there's these clinical partners as well that
Yeah.
potentially all have different expectations of what a joint project would deliver. And I really want to get your thoughts and kind of your experience on handling that kind of yeah.
Yeah, that's certainly true, Munya. You do have to talk to a number of potential partners to find the ones that you'll be able to work with. So, not laboratories have different pressures on them, and some laboratories are so busy that the idea of having personnel or time available to kind of assess emerging technology that comes across their path, it's just not in their remit. Like they don't have the time or the resources to entertain those kinds of activities. But then there are other laboratories where they will have they'll have a small amount of resources set aside for doing exactly that, partly because they want to know what's coming through in the pipeline. When it comes to replacing our existing systems, what are we going to put in there? And the only way they're going to get a better feel for some of those questions is to try out new technology as it comes along. So, we did talk to a number of partners. We found the ones that we work well with. It's a process. You just need to keep talking to people. You absolutely need to keep talking to people all the time.
Right. Cool. And I think in looking at the founding team for CytoPhoenix, I think there's quite a number of expertise, and I think that seems to be kind of a maybe secret sauce within the company. Could you maybe
Yeah.
speak a little bit about how you assembled such a diverse founding team that can kind of tackle various aspects of what you're trying to tackle?
Yeah, well, look, we started with a very strong microbiology-based team, and we had academic and clinically experienced people in that team. So there was already sort of 3 or 4 people there. And we knew that one of the big obstacles for microbiology laboratories, if they're using a technology like flow cytometry, would be the analytical part of the workflow. So microbiologists are not naturally flow cytometrists. So we needed to find a way to completely automate the data analysis. So the data handling and the data analysis just needs to happen seamlessly as part of the assay without microbiologist input. And we achieved that by writing some software that will do all that for us. So straight away, we had our own internal prototypes for that. But when we got serious about that process, we engaged a local software team from Perth, Western Australia, who had experience in getting software through the FDA and getting it approved. And we worked very well together on an academic slash commercialization type grant for 2 years. And that gave us an opportunity to get to know each other, to see how well we worked together. And that was an extremely productive project. It went very well. And we got sort of our first, genuine software prototype that would automatically handle and analyze the data. Then it was towards the end of that project where we all felt that it had gone well enough for us to spin out a company together, and CyTOPHENIX was founded at that point.
Oh, that's fantastic and a great story. So you're based in Australia, and I can imagine that, besides the time differences to many places, it might be challenging in terms of, starting up a company. But I think that there probably also advantages also being kind of within Australia. Would you mind maybe just speaking a little bit of some of the challenges and advantages that you've found about basing a company in Australia?
I'm certainly acutely aware of the challenges. So, you do things like podcasts at 9, 10, and 11 o'clock at night. No, you certainly have to be flexible with your timetable. I think I showed you that World Time Buddy is my new buddy. And you get very good at converting time zones into your own and vice versa. Look, you need to be very flexible. There's a fair bit of travel involved, not in the early stages of our company while we were really assembling a few other key pieces. But starting this year, we really began to travel more frequently to the major microbiology and infectious disease conferences because you need to be there. And so travel becomes a big part of your life as well. We've shared that out amongst the team members. Some team members have, they've achieved gold status on their frequent flyer points very quickly this year. Good luck to them. So you have to be flexible on time. You end up traveling a lot. Australia is a long way from other places in the world, and you really do have to engage very actively there's a fair number of agencies whose work is to promote awareness about antimicrobial resistance and about the ways we can tackle that, one of which is improved diagnostic tests. So exactly what we're doing. So we certainly keep in touch, via social media, with all of those agencies, and we attend lots of webinars online. You're actively building networks every day to reach out into the world and make the connections that we need to make so that then later when you are on the ground with these people, you already know them. You've already forged a relationship. So the tyranny of distance still exists, but it is easier to mitigate the tyranny and be ready for when you do meet in person. Yeah.
Right. Like maybe just to talk about on that grant that you mentioned that kind of allowed you to test these things out. How important are grants such as that that essentially allow you to start testing the water towards commercialization? Um, how important have they been to your journey?
Yeah, for us that it was really important. So, we had a number of purely academic research grants that, had put us in a position to make this discovery, and to characterize it well enough for us to think, gee, we've got something here. And then the inventors, they patented it. And then you're really starting to get into territory where the purely research grants, academic research grants, you may not fit the you're not necessarily going to be as competitive in for those grants as you were previously because you're trying to do your goals have changed slightly. We have an extremely accommodating state government who have worked very hard over the last 5 to 10 years to really increase the efforts to commercialise intellectual property in Western Australia. And so a number of grant opportunities specifically for that purpose, particularly in the area of health, have come online in the last few years. And there's been a lot of effort put into nurturing the ecosystem, helping people make connections, making sure that particular services are available, just lots of support to help energize the whole medtech sector. And, we've been commercializing this test during that time period. So that's been tremendously helpful to us and a number of other startups in Western Australia. There are also Australian funds that do similar things. So they tend to be even larger scale money. So, we're probably entering a stage now where we'll start to look at those opportunities and see where we might fit and what they can help us achieve. But there's just no question the state government support has been really beneficial to us. It gave us that first grant we met our software developers. We joined forces and founded a company. That's just straight up, that's evidence of how beneficial it can be when the right elements come together, as they did for us.
Yeah, great, thanks. And I can imagine that this is a very competitive space. I think you already mentioned a few kind of incumbents. Um, how do you deal with competition? There's some people who thrive on competition, is this something that just wanted to know, how do you deal with that? Do you just kind of maybe focus in on kind of what you're doing and really that is all you can really focus on, or yeah, are they things that you
Look, you certainly do need to keep your eye on all the competitors in the space, and there are a number of them in the infectious disease and antimicrobial susceptibility test space. We are quite fortunate in that our technology, we believe, unsurprisingly, that our technology um, offers some significant advantages over other approaches. So, flow cytometry, we know how powerful flow cytometry is as a scientific technique. It's used every day in hematology and immunology labs all over the world. And, one of the benefits of it is just the sheer number of cellular events that you can analyze in a very short period of time. And we're leveraging that power in our test. We test tens of thousands of bacteria at every combination of drug and concentration of drug and bug. Whereas some of the other emerging rapid AST technologies, they might measure a dozen, they might measure 100 cells, and then they move on and measure the next concentration. So when you measure as many cells as we do, we can be very confident about the data that we get from that and the answers that we derive from it. So, we're very confident that our technology offers some pretty significant advantages over other approaches. But it's not just the technology when you are developing a laboratory test, it's not just the technology. So we've got a strong technology. There are many other factors that we need to consider to get the test into the marketplace and being used.
right? I think as we wrap up, maybe just a few more questions. I think I wanted to know maybe what kind of advice, looking back now, would you maybe have for innovators that are building technology platforms out of university. I think that is one of my interests, and I think getting a piece of advice from you would be very important.
Look, I think putting yourself in a position where you are always open to the idea that something that's come across your academic research path might offer a solution to a problem in the world that needs solving. I think being open to that idea that you're going to encounter many potential solutions to problems, but getting the skill set together to be able to relatively quickly determine whether or not that genuinely is something worth pursuing in depth, or whether it's, yeah, it could maybe work, but this doesn't fit and that doesn't fit and that's never going to come together. So being able to sift through all of the ideas that you might have during an academic career to choose the targets that are more viable, more commercializable, more likely to succeed. So it's getting those skills that help you do that process. That's what's quite useful, I think, and doing that early. Yeah.
Okay, thanks. And there's this question that was asked a few seminars ago, and I've kind of put it in my repertoire of questions, where someone asked, what was the least natural aspect of being an entrepreneur or a founder that you found in your journey? So essentially, kind of, what was the least natural thing that you had to essentially become proficient at as you're kind of going
Oh,
through this journey?
gosh, I don't know. Ah, dear. Probably thinking or becoming knowledgeable about the in the end, someone has to buy your test. So who will your payers be? How are they going to get reimbursed? Who's paying them to pay for the test? So, thinking about some of those commercial aspects probably took a little more work than many of the other aspects of what we're doing. There wasn't really anything that stands out to me as completely alien that, I had to learn from the ground up. But that's probably one for which, it differs in each market that you go into, depending on how their medicine is organized in a region. And so you have to be able to pick up pretty quickly, who are your customers going to be? who's going to actually pay for the test, how are they going to afford to pay for the test. Um, that becomes a whole enterprise in its own right. Yeah.
Great, thanks. And then lastly, kind of, looking forward, what excites you most about kind of where Cyto Phoenix is going, or what they're currently doing, what you're currently doing now?
Look, it's one thing to notice something in an academic laboratory and think, gee, that could be really useful in the world. It's another thing to actually get it out into the world where it can demonstrate its usefulness and create impact. So, at the moment, our test is a fantastic test that has the potential to save many lives, enhance outcomes in many different ways, but that is never going to happen unless we commercialize it and get it out there and in use. And I think it's the fact that we are the people doing that part, getting it out there. That is super exciting, because when that clinical impact happens, that's going to be because our team did what we did together.
Great. No, thanks for joining us today. I will leave it open for a few questions, maybe from the attendees, if there's any questions from the attendees. Okay, does that look like there's anything that's popped up? So again, thank you again, Christine, for taking the time to chat with us and share your wonderful story. And that concludes the November Innovation Stories. And have a good night, and yeah, enjoy the rest of your week.
Thank you.
Thank you. Bye, everyone.